Hello, my name is Shanika Jackson, and I am a senior at School Without Walls High School in Washington, D.C. The title of my project is Genetic Variation versus Environmental Contaminants in Striped Bass in the Anacostia River. My hypothesis goes as follows. The null hypothesis states that there is no correlation between striped bass, genetic variation, and endocrine disrupting chemicals, also known as EDCs, and the alternative hypothesis is there is a correlation between striped bass, genetic variation, and EDCs. So, to start, I interned at Georgetown University with my mentor, Dr. Matthew Hamilton, who is a population geneticist as well as a professor at Georgetown University. He and a couple of other folks in the lab that he helps runs really helped me hammer out the details to my project and understand what I wanted to do. The basis of my project was really trying to see if certain chemicals called endocrine disrupting chemicals were mutating parts of the genome in striped bass. And it wasn't always striped bass, it wasn't always aromatase B, which I will get into more later. But the people there were really supportive and really helped me understand exactly what I wanted to do with my project. So the main tool that I used in doing my experimental design was PCR, or polymerase chain reaction, and it's a tool that's used to amplify millions of copies of a specific part of the DNA. And the DNA, you can use it for sequencing after you actually get the primer design right, and or you can do a lot of this PCR. But I wanted to use it for sequencing. Again, I use special primer design for striped bass. Primer design is like pushing Control F in a long text on your computer. Because we can't exactly see what's in the genome, we can't just go in and find it. We have to send in special tags or primers, and they find it for us. This is a very hit or miss process. My teacher really told me when I first started, it's a little bit of science and a little bit of witchcraft. And my mentor told me it's hit or miss. And I really understood what he meant when I actually did it for myself. It took me a good 4 months to design successful primers, and that's because my mentor is really old school and he made me do it by hand, even though there are a lot of software programs that do it for you. But I'm grateful because I learned a lot in the process. Also, I looked at Aromatase B and striped bass. And like I said, striped bass wasn't my first choice. I really wanted to look at smallmouth bass because I interviewed Dr. Vicky Blazer earlier in the year, and she told me about her work with smallmouth bass, and I wanted to continue it, kind of challenge her research, not in any way as official as and efficient as hers, but I really wanted to give her something else to think about for her studies. And aromatase B is the gene that I was looking at while I did the primer design in the PCR. It's a gene that converts androgens into estrogens in fish and also other organisms, but I was specifically looking at fish. Before I go any further, I would like to explain what an endocrine disrupting chemical is. It's any chemical that has the ability to disrupt the signal pathway of the chemicals of the endocrine system, which controls hormones. And the ones that I was looking at specifically was sex hormones, testosterone and estrogen. So my findings, my PCR and my primer design yielded inconclusive results. The way that this works is that you design your primers, you run a PCR, and then you check your results by a gel electrophoresis, which is like gel that you actually plug into an electric current and runs the DNA for a good 45 minutes that I did, and you see the different pieces of DNA and see if that's what you were looking for. I did this 3 times and it gave me inconclusive results, so I couldn't go on with the sequencing like I wanted to, but I was very happy that I actually amplified aromatase B. Aromatase was around 1,200 base pairs, so it was really big, and I was very surprised that I actually got any result at all, so I was very happy about that. I had a lot of limitations. The bass that I was looking at were not from the Anacostia River. They were actually from the Patuxentine River in Maryland, and but the Anacostia and the Patuxentine River are both a part of the Chesapeake Bay, so they're exposed to the same things. Also, all the necessary tools were not available for my project. I wanted to correlate the endocrine disrupting chemicals and the levels or the concentrations in the tissue samples to more variation in the genome if I was able to sequence, but I was not able to. One, I was not able to have a successful primer. And also, it was very expensive to get chemical testing. Lastly, I only had under a year to do this research, and while I thought that was a long time, it's really not when you really start working, and I'm very conscious of that now. If I was to expand my research, I would get or look at more genes to test for the variation. And because I only looked at one gene, I would look at maybe 5 or 6 more. I would also look at more fish because I only did focus on this one fish. But aromatase is a gene that's found in all all teleost fish. And I was also very interested in the sex determining systems of fish. It's really complicated, complex, and intricate, and I really loved reading about it and researching it. Also, I would really try and do the chemical testing I was talking about. I think that would make for a much stronger correlation. Thank you so much for listening. I hope that my project was just as exciting as it was to me. As to you as it was to me. And thank you for listening.